So thanks everyone for having me today. I'm very glad to be able to tell you a little bit about what we've started learning about congenital diaphragmatic hernia. So for those of you who don't think about CDH or congenital diaphragmatic hernia all the time, this is a congenital anomaly that we see in terms of a defect in the diaphragm. The diaphragm is your breathing muscle, and it also separates or acts as a barrier between your abdomen and your what we call your thorax or your chest cavity. When babies have a defect in that diaphragm as they're growing in utero, what I'm showing in the figure on the right and that sort of pink stuff is that their intestines can actually move up into the chest and can compress the lungs and the heart and can cause problems in terms of lung development. And we see some of that postnatally. We see this congenital anomaly about 1 in 2,500 births. And unfortunately, it can result in a very high rate of mortality of about 30% because you can imagine it's difficult for a baby to breathe, and if this is not recognized early, can in fact cause those breathing problems. It can be associated with other things as well. Congenital heart disease is one of the things that we see most frequently, and I'm going to come back to that, but I think one of the real important things about the Gabriella Kids First program is the ability to look across birth defects, and that's one of the things I'm going to emphasize today as we're going through this. As we started looking at this now over a decade ago, there was some evidence that this might have a genetic component because we knew there were some children who had chromosome issues. We also knew of some rare genetic conditions with what we call single gene disorders, and there were not many, but cases of twins where we could see it running in twins or within siblings or even within families from parent to child. We also knew in some cases of animal models, in particular mice that had diaphragm defects in development. So as we did this, we put together a consortia of many different clinical sites around the country. You may see some of your local institutions. We've also since expanded, so we now have international sites around the world. And the point is that for relatively uncommon anomalies, we need to be able to put our heads together, and that's what we've done. As we've done this, we've had really wonderful families that we've been able to work with, and one of the things that we've done is not just studied them at birth or the time they joined the study, but continued to follow them as the children have been growing up. And I have to say personally, as a pediatrician, that's been a great thing to do, is to watch the kiddos grow up. As we do this, we've taken many, many different data points or clinical information that we did about how they did in the neonatal period, how old they were when they went home, if they had other congenital anomalies, if they had other medical issues, if they were having trouble growing. One of the things we can see, as I said, is breathing problems. So we looked at that as well as an associated condition called pulmonary hypertension. We actually had all of our echocardiograms read by a single center so that we could have consistency in assessing the level of pulmonary hypertension. And as we followed the children growing up, we've been following to see how they've been doing in terms of growth as well as development. So we've actually done some, I think, consistent, rigorous, standardized methods of assessing development at 2 and 5 years of age, which have been helpful for us. When we started this a long time ago, we actually started out by first using some blunter genetic tools out of a karyotype and chromosome microarray, and have gone on to exome, and then with Gabriella Kids First, gone on to genome sequencing as well. As we did this in the early days, as I said, there were some things that we could see by looking at kind of a high level. Chromosome conditions like trisomy 21 or Down syndrome, some translocations, as well as copy number variants. And just to give you a sense of what this looks like, they're distributed throughout the genome, so they weren't located in any one particular place. But I'm going to show you one circumstance, and I'm going to point out that we saw in particular a region on chromosome 8, 8p23.1, where we saw not just one individual with CDH, but actually a series of individuals with congenital diaphragmatic hernia that had regions that were all in 8p23, but they were varying in terms of size. And we could look at what we called a critical interval, the one that was shared by all of those individuals. And I'm showing down below the different genes that we saw within that interval that was deleted. And importantly, I'm going to draw your attention, I show here GATA4 as one of those genes that we saw within that interval. Again, it wasn't the majority of individuals with CDH. We certainly have not found one single gene for CDH that explains all of it. One of the bottom lines is that it's genetically complicated in the sense that there are many different ways at which we can disrupt diaphragm development. In the early days, after we were able to do that, we couldn't quite afford to be able to do genome sequencing, so we took a shortcut, not just we, but many others in the field. Your genome looks something like this. It looks a bit confusing and difficult to decipher, but if you know how it's organized, you can see some of these gold nuggets, as I'm showing here in terms of being able to see the Gettysburg Address of 7 years ago. And the point is that if you can focus on those gold nuggets of the so-called coding regions, or the portions of the genome that encode the proteins or the gene products that our cells make, then you can cut down the workload to about less than, not quite 1/100th, but a little bit more than 1/100th of the work that you would have to do. And so that was an important advancement in terms of our understanding. As we started doing this, one of the obvious things to do was to look at our families, where we saw more than one family member with diaphragmatic hernia, thinking that there might be a greater likelihood of something genetic if we saw it running in a family. So I'm showing you here one of our families, and colored in black are the individuals with diaphragmatic hernia. You can see the arrow in the bottom row is pointing to the child who actually was enrolled in our DREAM study. That child had an uncle, so his father's brother had passed away actually when he was just a little baby of diaphragmatic hernia, so that we knew that we had an uncle-nephew pair in terms of doing that. So we sequenced, in this case, the young man with the arrow, his mom and his dad, and started looking for what might be something related perhaps to diaphragmatic hernia. As we did that, we saw that he actually had a genetic variant, one that we call a missense genetic variant, or a single amino acid substitution, one single building block within a protein that was swapped out. Instead of a red block, it was a blue block. So it could be thought of as a somewhat subtle change. But I want to point out to you that that gene that was affected was that same GATA4 gene that I showed you back in the chromosomal region of 8p23. So before we were seeing that it was just deleted, that in several individuals we were missing that gene. Now we're seeing a more subtle type of change, again, something that would change the GATA4 perhaps so that the binding affinity for this transcription factor, or this gene that orchestrates and organizes the expression of many other genes, might be affected. So as we did this, again, we stopped and thought to ourselves, does it make sense that this might be the gene that's involved? And we thought logically it made sense. This was a gene that is expressed in the right time, in the right place. We knew it had been associated with congenital heart disease. We know mice, or at least some mice that had mutation in this gene, also had diaphragm defects. So it made sense to us. But we were starting to ask ourselves whether the specific change that we saw in amino acid 252, in fact, whether or not that was problematic. So we went back to our family and said, well, in fact, if it is, we would expect it to be traveling with diaphragmatic hernias in the family. And I've shown you here again, I've added the genetic results for the family. So what I mean to show here is anyone who has a minus sign is carrying the genetic change that we see in the GATA4 gene. And you can see that the young man, he has a minus sign. You also see his father also seems to carry the same change. And his grandfather also, we saw, carried the same change. We didn't have DNA on his uncle who had the CDH, or the diaphragmatic hernia, so we couldn't test him directly. But we did test other people in the family who didn't have any of the diaphragm defects, and they didn't have that change. But I remember being a little bit puzzled and saying, but I don't understand. His father seems to have this change. His grandfather seems to have this change, yet we don't see any diaphragm defects within them. How could this be? Maybe we have the wrong answer. Maybe we need to rethink this. Or maybe I thought we switched the samples. Maybe we accidentally, sequenced the wrong person or made some mistake or had some contamination. So we went back, we checked all of those things again, and then it sort of struck me one day as I was, believe it or not, waiting in a very long line and just sort of daydreaming, having my mind wander. And I thought to myself, well, maybe his father really does have a diaphragm defect and we just don't realize it. We haven't really looked very carefully. Maybe we need to go back and look. So we did that. We went back to his dad and to his grandfather, both. And these are MRIs, ways of being able to image and look very closely at the diaphragm. And again, they weren't complaining of any symptoms or any problems. But if you look at each place, I don't think you can see my arrow on the screen. But if you look at each place where the arrow is showing on the MRI, there are 4 of the arrows in the 4 different panes. Each one of those is actually a very small defect in the diaphragm where you can see things either bulging through just a little tiny bit, not enough to cause any problems with breathing, not any problems in terms of lung development, but clearly a problem in terms of how the diaphragm developed. And actually, since then, since we had this first realization, it's something that we've done repeatedly and realized that in some families, there are individuals who have very, very subtle changes that tell us that there is really a problem with diaphragm development, but thankfully hasn't been so severe that they needed to have surgery. So beyond that, we went to be able to see whether or not we might see genetic changes, but in a different family structure. So most of our families actually were not familial in the sense that there were 2 or more people with diaphragmatic hernias. Most of our families, there was only one individual with a diaphragm defect. And again, if you think about what I was talking about before, this is a really serious birth defect. If people didn't appreciate it, if you didn't get diagnosed quickly with this condition and make it to the right hospital with the right surgeons, really a generation ago, most individuals, most babies that were born with diaphragm hernias actually died from their condition. And when you think about that, if you think about if those individuals died, what happened with those genes? I mean, their genes died with them. How would they have been passed down in the families from generation to generation? It could have been as in the family I just showed you, but in many cases, they wouldn't have been passed down. So we thought to ourselves, maybe in terms of where these genes come from, maybe they're de novo mutations or new mutations in the families, and that's how they arise. So we started looking at that and looking at many of our families where we didn't have multiple people with diaphragmatic hernias. And we saw that about 6% of them had these copy number variants I alluded to before, and about 20% of them that had CDH plus something else. We could see what we thought to be a damaging or deleterious de novo genetic variant. And even the ones who had just an isolated CDH, although it was a lower percentage, it was only about 10%, about 10% of them had these de novo changes as well. So I'm going to do just a very brief primer because I know some of our families are watching today. So just so you understand some of the gobbledygook that we talk about. Having these de novo genetic changes is not specific to individuals with CDH. In fact, I can tell you I've done the experiments with my own 2 sons. We've looked in terms of my boys and looked at their genes compared to my husband and myself, and they literally have a little over 100 genetic changes that are different from either of us as their parents. They don't have diaphragm defects, and part of the reason is because they got lucky to a certain extent. Those genetic changes that happened anew happened to fall in places that didn't matter so much. One of the reasons is because I'm showing you here that for instance, this type of genetic change where you change this alphabet letter of a C to a U, it doesn't happen to change that amino acid in the protein. We call it silent because it doesn't change the amino acid. We all have many of those, and they don't cause us any problem. They don't change anything for our bodies. Some other changes are what we call missense. Changing this G to a C changes this genetic code for the amino acids, and it changes that genetic code from one of those building blocks called an alanine to another building block called a proline. Many of those changes are benign or not problematic. We have ways of being able to predict now better which ones will or won't be a problem, but many of those are just fine. Our bodies tolerate them without a problem. But there are a class of changes that, for the majority of individuals, cause problems. And again, it's the same type of change in a way. It's just a change of one alphabet letter of an A to a U. But in this particular case, it causes what we call a premature stop. Instead of making the full protein, it makes only part of the protein, and then it stops making the rest of it. And in many cases, in fact, probably the majority of cases, those disrupt the protein and how that works. So I gave you that lesson because as we've done this, we've looked at, from a statistical point of view, the frequency that we saw such changes in our individuals with CDH, and to be able to look at them by these different classes. So again, in doing these and then also looking at individuals without any of those congenital anomalies as a reference or as a comparison, we started looking at the frequency or the enrichment of that. So those silent changes that we talked about, we looked at different types of things. The black is showing all genes. The pink is showing a class of genes, about 2,000 of them that we call constrained, or they're kind of rigid. They like being just exactly the way they are. And then these in light blue here, other genes. And when we look at these silent changes, these ones that don't change the protein, we don't see really any difference. This dotted line here of 1 means that the ratio or the comparison is about 1 to 1. Again, no more, no less for individuals with CDH than for other individuals who don't have any congenital anomalies. On the other hand, if we look at the group as a whole of all of the missense variants, we see a little bit more. Statistically, these are error bars here. Statistically, it still is right near 1, so we don't statistically see a difference here. But now when we take the category of deleterious missense, or those that we have ways computationally to predict might be causing a problem, for those individuals with CDH, we see an enrichment or an increase in the frequency for all of those, either in constrained genes or other genes. And then in particular, if we look at the likely gene damaging, we see a much higher frequency, especially in this pink dot here for the genes that we call constrained genes. So we're starting to hone in on the signal. It's not telling us yet necessarily which gene within all of these is causing the problem, or which multiple of these we're going to be able to clear as CDH genes, but it's telling us that we're on the right trail. As we did this, one of the other ways, we looked at it in a couple other dimensions. We did have the sense, as I was alluding to before, that if you had not just diaphragmatic hernia, but might have another associated birth defects, something I've called a complex case as opposed to an isolated case, that there might be a greater likelihood of having a genetic or at least a single gene effect in those cases. And again, what I'm showing you here is the complex cases on the left of the side in blue compared to the isolated cases in red. And again, you can see there's a higher frequency of those for the deleterious missense, and just slightly higher, I'd say really about the same, for the likely gene damaging or disrupting variants as well. Now, interestingly, we were also looking at the differences by sex, whether or not there was going to be a similar frequency in boys or girls. We do know that CDH is slightly more common in males than it is in females, almost as if females are somehow protected in a way from this, or males more susceptible, depending on if you look at the glass half empty or half full. And so when we were looking again at these de novo variants, we saw in pink in the girls, we saw a higher frequency of these de novo deleterious missense variants or these likely gene disrupting variants, either one of those. It seemed as if for these girls, they needed to have what we think as one of these factors that was disrupting the development of the diaphragm that would have caused the problem for them. And the way I'm showing this is to be able to think about this as a threshold of liability, as one of the ways that we think about it, that again, girls might be somehow protected. They have a higher threshold in this. Boys, for whatever reason, might have a lower threshold. And for these girls, it really takes a big sort of something pushing them hard to be able to get over this threshold. And we think that some of these single genetic factors, in fact, are very sort of impactful, have a very high risk associated with them. In contrast, what that may be telling us is that there are multiple genetic factors, each of one of which may not be as strong, that are accounting for more common, more commonly for individuals with diaphragmatic hernia. So one of the next things we did was to go on with the Gabriella Miller Kids First funding to be able to look at whole genome sequencing. That is to look at everything, not just the coding regions, but the non-coding regions as well. And as we did this, an interesting pattern started to emerge, not just because we could do whole genome sequencing, but because we could actually begin to look across congenital anomalies and start to see some patterns. And so we started this story with CDH, and people in my team thought I was dyslexic because the next thing we did is we looked at CHD, or congenital heart disease. And what we realized, and I put these in two columns here, one under diaphragm defect, the next one under heart defect, we saw, in fact, that we were seeing these de novo genetic changes in one particular gene, MYRF, in both individuals who had either who had CDH, and many of whom also had CHD. Interestingly enough, it was not just those two congenital anomalies, but you'll see in the column called congenital defect. Some of them also had other genitourinary malformations. We had some of the babies who had ambiguous genitalia, some who had undescended testes as something that was milder, or in fact having no internal genitalia. We also saw that in some cases they had some other anomalies associated as well. So it looked like this was a gene that was having effect in multiple dimensions. I'll also show you that in the status column, unfortunately, many of these children didn't make it. Many of them were severely impacted, especially if they had both congenital diaphragmatic hernia and congenital heart disease. And so we were starting to also get some information in terms of prognosis. I'll also point out one unusual thing that has taught me a lot. Patients always teach me a lot, I have to say. We had one set, you may see it about 2/3 of the way down, called female identical twins. So we had one set of identical twins. Unfortunately, neither one of them made it. But even though they were genetically absolutely identical, and we've shown that with their sequence, one of the girls had a diaphragmatic hernia. The other twin had hypoplastic left heart syndrome, a type of congenital heart disease. And it just shows to me that even though they had the same genes, the same womb, the same in uterine environment, they both had certainly a manifestation of this, but not the same one. And so there are other factors beyond just the genes, even just the environment. And so what is this gene, MYRF? It's also a transcription factor, the theme for the day in terms of this. It was originally thought, and it was named after myelin, or a brain protein. It was thought to be mostly involved in terms of brain development, but I think we're just realizing that that was in some ways a misnomer, and I think if we had to rename it, we probably would name it differently because it seems to have a much more global effect in terms of body development overall. With the Gabriella Miller Kids First data, we're also going on to look much more systematically at the non-coding variation. So far, what we've seen is that we again see many new genetic changes within that region. We're starting to get a sense of what the impact of that is, but to say, I would say the short story is that it's complicated, and the impact of the things that we're seeing in the non-coding space is not as impactful as, at least per person or per genetic variant, as some of the variants we're seeing in the coding space. So there's more work to be done. So just to wrap up and say in summary that as we've done this, and just to give you a sense of this for any of the families who are listening in on this, this was really a whole community effort in terms of doing this. We literally have analyzed hundreds of families who have all worked together with us in doing this. And by doing this, we have started to chip away to understand that, importantly, something probably like about 15 to 20% of families now with CDH. We have, I think, a very good idea of what's going on that's causing their diaphragmatic hernia. I think we have an idea about some other genes that are contributing, and I won't have time to talk about it today, but we're also using this to be able to check this out and see if we're right by making mouse models of this to test our hypotheses. Most importantly, I just want to end by saying that we have a fabulous team driven by the families who really are, as I said, our partners within this. I want to put out a shout out to Julia Wynn and Rebecca Hernan, who are coordinators who've done a fabulous job with this. And just to say that it really does take a large team, fabulous team of dedicated surgeons, geneticists, neonatologists, gastroenterologists, but I'll stop here and be glad to take questions. And also I should acknowledge our funding, which has come from NIH as well as many of the families.